This is the second screencast in the muscle physiology series. We're focusing in this screencast on tissue organisation of skeletal muscle. You'll notice that the diagrams that we're going to show you in this screencast are all very similar. They're all taken from the same textbooks just for consistency. What is important here is we're going to give you lots of terms. I'm going to tell you about the individual structural components of skeletal muscle. Probably in an examination, we wouldn't expect you to necessarily remember all of these terms, but what we really want you to do, as in all of probably PM1A, is to be able to relate the structure to the function. So we'll focus on what these elements are, why they're important for the function, in this case of skeletal muscle. So first of all, you'll notice that this is a diagram maybe that's related to the diagram of the muscle fibers, in particular the red muscle fibers that I've shown you on in Screencast 1. We'll see that things like blood vessels and different connective tissue layers are labelled in a little bit more detail on these diagrams. What is important is really to go from the whole muscle organ down to the individual filaments, and I'll draw your attention to that when we get to the relevant slides. So this is like a nice diagram just showing you maybe how the muscle organ is attached to the skeletal muscle framework via these tendons, and starts to introduce some of the different types of connective tissue and how the muscle is organized in order to fulfill its function. Skeletal muscle has two ends, typically attached by tendons to the bone. And again, we talked about how tendons have receptors that control our sense of where we are in the world, our proprioception, and muscles also have organs or have receptors that control proprioception. You can look back into the previous screencasts on the nervous system if you want a little bit more information about that. What this diagram does do is illustrates there are three main types of connective tissue. Maybe on the outside, the epimysium, more intermediate connective tissue called perimysium, and the endomysium, which surrounds these individual muscle cells or the myofibers. It also introduces this terms of fascicles, and the fascicle is effectively a bundle of nerve cells. So you can see that these nerve cells are arranged in bundles. The original term actually comes from bundles of wheat that you could group together. So that's what a fascicle is if you see that term. The three connective tissues, the epi, the peri, and the endomysium, are continuous with tendons, and they bind the muscle cells together. So this again shows you a muscle cell. Maybe it's kind of an intermediate diagram between the neuromuscular junction, where we've already shown you things like the sarcoplasm, the sarcolemma, T-tubules, and some of the other elements like the terminal cisternae. This really shows where these terms fit into that previous diagram about general tissue organization. So we already know that the sarcolemma is the plasma membrane of skeletal muscle. We've described these T-tubules. They're not so obvious on this diagram. They're kind of shown here. We saw on the previous diagrams, and we'll come back to they've got this nice T shape. Probably important for excitation, contraction, coupling. Some of the proteins that we're going to introduce you to are arranged around the T-tubules and are important for contraction. Similarly, these terminal cisternae, which we already introduced in the previous screencasts, we said were calcium stores that were important for muscle contraction. They're arranged near the ends of the T-tubules, and there are specialized forms of these reticulum intracellular calcium stores. We've heard about endoplasmic reticulum maybe in other lectures. These are called sarcoplasmic reticulum, SR. Anything with sarco in front of it pertains to muscle. So the terminal cisternae are just a specialized form of calcium store, specialized in the muscle form of sarcoplasmic reticulum, SR. The triad is a region where one T-tubule is flanked by two of these calcium stores, two terminal cisternae, And myofibril is a bundle of contractile filaments within the muscle fiber. Again, we'll see how myofibril fits into the overall description of muscles when we consider the overall organization. And this is maybe the first diagram that shows you these filaments in a little bit more detail. So we have thin actin filaments and thick myosin filaments, and they're arranged in here. We'll give you a little bit more of a focus diagram on the next slide, but they're arranged, the way that they're arranged forms these lines that we see. So if you look, basically if you looked at a microscopic image of a muscle, you would see these different lines. This is a definition of the lines here. I think, you know, take home message here is, in an exam we wouldn't ask you what an A band and an I band was, but maybe we would like you to know how these different lines are formed. They're formed basically by the interaction, or the way that the actin and myosin, actin in blue, myosin here in this kind of color, we'll see these in a little bit more detail, are formed within the muscle. Maybe one important term is the sarcomere. That's the contractile unit between these Z lines. And again, we showed you this on a diagram of the neuromuscular junction, and we'll come back to that in the next few slides. So just be aware of these terms, don't necessarily commit them to memory, just know what they do essentially. So yeah, maybe this is a good diagram to focus in a little bit. This is the thin actin filament. Hopefully you can see the difference, or the reason we call it thin compared to myosin, which is a thick fiber. Again, in the next screencast, we'll show you a thick filament. We'll show you why this is a thicker filament, and maybe the importance of these myosin heads in particular for excitation contraction coupling in the sliding filament model. Yeah, I think if you were going to focus on one diagram out of all of these, just to show you the overall tissue organization from muscle organ down to individual actin and myosin fibers, this is an important one to know. So essentially what we're doing here is we're going from a gross organ level down to the individual components. This is the muscle organ, made up, as we've said, of these different connective tissue components, epimysium on the outside, arranged in these bundle or fascicles. So this is the muscle composed of many of these fascicles. The fascicle itself is composed of many myofibers. So you can see this is the fascicle, these are the myofibers, the muscle fibers, the cells. And again, we can see this maybe intermediate connective tissue layer, the perimysium. The muscle fiber, the myofiber muscle cell is composed of many myofibrils. So you can see that we're getting further and further down in terms of organization. So the myofibril is an individual unit here. Many myofibrils make up a myofiber. And if we look down at the individual myofibril, we can see that they're composed again of an individual element, the myofilaments. And the myofilaments finally are the thick myosin and the thin actin. So again, take home message, if you go from the bottom up, this is how these filaments make up a fibril, make up a fiber, make up a fascicle, make up a whole organ. So just be familiar with some of these terms. So if we're thinking about skeletal muscle, we introduced this term, we introduced the term muscle unit in the previous lecture when we were talking about neural input. We told you that skeletal muscle is voluntary, that it requires nerve input innervation, so it's neurogenic. And remember that we said that cardiac muscle and smooth muscle is myogenic, so it's different in terms of requiring neural innervation in order to contract. We told you before that the nerve that supplies an individual, or nerves that supply skeletal muscle, are called alpha motor neurons, and some of the differences between these and other neurons that we considered. So this is maybe an important bit just to clarify. Each adult myofiber that we saw on the previous slide is typically innervated by, or typically is innervated by one axon terminal. So there's a one-to-one relationship between the axon coming in and the myofiber at the neuromuscular junction. By contrast, and hopefully this will become clear over the these couple of sentences here, a motor unit is defined as a single motor neuron and all the muscle fibers that it activates. So importantly, while the relationship between a myofiber and an axon terminal is one-to-one, the motor neuron is defined as all of the neuromuscular junctions effectively that control a muscle. And hopefully you can see that if you've got a small muscle, something like the eye, you've got a small motor unit, whereas if you've got a large muscle, something like the quadriceps in the leg, you need much more prominent supply, so you've got a large motor unit in order to be able to contract a larger muscle for strong contraction, it says here. And again, if this is maybe not clear, hopefully we can just focus in just with a couple of reminder slides. We've talked about the neural side. We've talked about how the nerve innervates. If you were wondering before what the endomysium was when we talked about the neural inputs, now hopefully you know from those previous slides and what a muscle fiber is. But the thing probably to focus in here is on the motor unit. So we've shown this one-to-one relationship here between the myofiber and the axon, myofiber there and the axon, but a single motor unit may have between 3 and 1,500 muscle fibers. So 1, 2, 3 is shown here. So this is a motor unit, this is a one-to-one innervation, this is a one-to-one, this is a one-to-one. So just make sure you know the difference between a motor unit and individual innervation at the neuromuscular junction. This is a diagram that we showed you before. This was obviously, we've talked about how acetylcholine is released across in this case the synaptic cleft that is incident on a motor end plate, and we showed you the terminal the T tubules and the terminal cisternae, the Z line and the sarcomere before. This is what we described about exocytosis. I won't go into that again. You can look back on the neurobiology, nervous system lectures. What we'll do here is we'll just focus on these final few points before we finish this screencast. So the muscle action potential spreads across along the sarcolemma, this plasma membrane of the muscle cell, to the T tubules, triggers calcium release from the terminal cisternae. Remember that these are specialized parts of the sarcoplasmic reticulum. And that is the final trigger that couples excitation to contraction. Contraction is due to a number of calcium dependent proteins. We'll talk about these in the next screencast, but this is just to summarize it. Calcium binds to a protein called troponin, which is present on the actin fibers, permitting cross-bridge formation, or changing the cross bridges between the actin and the myosin, and the muscle contracts according to the sliding filament model, and that requires ATP. As I said, we'll cover this in more detail in the next screencast. Just to complete the cycle, if you like, this is really related to neurotransmitter information, but acetylcholine is continually degraded into acetate and choline by this enzyme acetylcholine esterase at the neuromuscular junction. The calcium levels fall within the muscle and the actin-myosin interactions are inhibited and the muscle relaxes. So that's a good place to finish this screencast. In the final screencast, we'll talk about excitation, contraction coupling, and in particular, give you some information on the sliding filament model.